Hi, everybody. I think we'll start. Good evening, and welcome to our fourth and final talk in this Wednesday of the Genome lecture series. I truly hope you enjoy the first three talks, and I'm absolutely sure you'll enjoy the talk today. And so with that, let me just introduce you to our speaker, Dr. Evan Eichler. Evan is really somebody who doesn't need any introduction. In fact, we consider skipping the introduction altogether, but if you've done any kind of research in genomics or structural variation or autism, not only you know Evan, but you probably read many of his papers. But because I know some of you are not coming from genomics, let me still tell you a few things about his background. Evan comes to us from the far, far land of Canada. He did his bachelor degree at University of Saskatchewan, probably butchered that, and then moved to the U.S. to do a PhD at Baylor College of Medicine at Houston and a postdoc at Lawrence Livermore National Laboratory. He first took a faculty position at Case Western Reserve University at Cleveland, Ohio, before realizing Seattle is a place to be. Moved to the UW, where he's now a professor of genome sciences and investigator in the Howard Hughes Medical Institute. If you're still not convinced that Evan is a wonderful researcher and a wonderful speaker, let me just tell you he's also an elected member of the National Academy of Science, an elected member of the National Academy of Medicine. He won more awards than I can list and published, last time I counted, more than 370 peer-reviewed papers. And if you're still not convinced, let me just share with you one quick anecdote that I still remember from my first week here at UW. As you may know, one of the challenges as being a junior faculty is to find really good postdocs. And I remember talking to another junior faculty who told me he was just interviewing a really wonderful postdoc that he really wanted, but he ended up going to Evan's lab. And then he ended up saying, well, but losing to Evan is okay. I can live with that. And so I hope that with that you're all convinced and are all excited about the talk. So please just help me to welcome Dr. Evan Eichler to the podium.
Well, thanks, Elhanan, for that probably too generous introduction. So it's a real pleasure for me to be here today to share with you the research that we've done in my lab over the last, I guess, 15 years. So it's going to give you a perspective that will involve work that I started before I came to Seattle and work that we've really continued up to really the present day, with a specific focus on trying to understand the genetics of autism. So let me start with essentially what autism is. So autism is generally recognized as a neurodevelopmental disorder, at least that's how we perceive it. It's characterized by typically speech delay, impaired social interactions, and repetitive behaviors. So if you've ever met an autistic child, you'll know they often have very repetitive behaviors, stereotypic movements. It's important to note that not all children have all features, but they have to some degree impairment in these three areas. The estimates that came out as of April this year from the CDC estimate that 1 out of 59 children actually have autism. So this is an increase now from the 1 out of 68 that was reported 4 years ago. It's important to note that autism, at least the definition of it, and it's now referred to typically as autism spectrum disorder, is an umbrella term. It encompasses many different types of features, and it's an umbrella that I would argue that has gotten bigger over time with increased awareness of the disorder. And also relevant to this talk today is the fact that children with autism often share other features with other disorders. So often, but not always, about 40% of the kids with ASD or autism spectrum disorder have intellectual disability. They're susceptible to seizures. So many of them have a diagnosis of epilepsy. As well as when they get into adulthood, there's often, even within the families, there's association with neuropsychiatric conditions, such as schizophrenia, bipolar. So they tend to run together when they are in families. So this talk today is about the genetics of autism. And let me kind of assure you that, in fact, the data has been really consistent, despite a lot of hoopla 15 years ago about other factors, even though that we believe they're environmental factors. We know that the genetics is a strong component. So the current estimates suggest that as many as about 75% of autism has a genetic component. And some of these studies come from identical twins, where people have looked at essentially identical twins versus twins that are not, or individuals that are not identical within a family. And when one child is identified with autism in an identical twin, the chances that the other child will be diagnosed with autism is close to 75 to 90%. Within families, the recurrence risk is not too high, so it's essentially between 5 to 20%. But that means that if the population background is about 1%, there's obviously a genetic component, for some families at least in terms of risk, that runs within the family. A really interesting feature of the disorder is the fact that males are more likely to be affected with autism, and we're going to come back to that today. We have some insights from studying the genetics of individuals with autism, but current estimates about 4 males for every female diagnosed with autism. And if you go to high-functioning autism, which used to be referred to as Asperger's, that number is closer to 6 to 8 to 1. So a real bias for males in the human population over females. When I came to Seattle, there had been quite a few papers looking for common genetic variants underlying autism, and in fact, most of they came up empty. People argue today that that's because the initial studies were underpowered, but people thought because this disorder is so common, maybe common genetic variants really play an important role. We knew from studies that I had even done when I was at Baylor College of Medicine that we would actually have sometimes disorders that would run within families. But that explains really a relatively small fraction. It is not the majority of families. Most of the families that have autism, we call them out of the blue, because there's in fact no family history of autism in those families. So if there's a strong genetic component, we know that 70% or roughly 70% of autism has a genetic component. Where's the rest? So I want to share with you kind of a little bit of my background and my motivation for why I've been particularly interested in this. There are a few reasons, but one of the things that I did when I was at Baylor College of Medicine, I focused on trying to understand the genetic mechanism behind one of the most common causes of developmental delay in the human population, also associated with autism, and that's shown here to the right. These are children. Does anybody recognize what that disorder is to the right? Okay, well, let me ask you a question. Does anybody recognize the disorder to the left? Down syndrome. These two, this is Down syndrome, and when I came to Houston, this was just discovered. This is Fragile X syndrome. These two are the most common forms of intellectual disability in the human population, and about 40% of these kids, or 30%, I think is the last estimate that I saw, also would be classified as autistic. Now, these individuals tend to not be autistic, although there's a fraction of them that have features that are reminiscent of autism. So when we were in Texas and we were screening and identifying the gene responsible, most of you still haven't heard of Fragile X syndrome, but in fact, it is the second most common form of intellectual disability in the human population, and the chances are you have seen someone or met someone with Fragile X. It's just that it wasn't actually recognized as such until 1991. So the motivation comes from this. When we were doing these studies with children with autism and developmental delay, most of the children that we believed there was a genetic cause came back after doing workups, looking at their chromosomes, or workups looking for known genes, came up with no genetic diagnosis. So back in 2000, the estimates were that 75 to 80% of children with developmental delay, and the numbers were even higher for autism, had no genetic diagnosis, even after multiple testing was done. So one of the motivations in our lab is to work on understanding the genetics of autism. Why? Because in terms of the families, understanding why their child is the way that it is incredibly helpful, even if there isn't any treatment or cure immediately available. We know that once we understand the genetics, we can inform individuals in terms of diagnosis and counseling. So a big question that often comes up in families, if they have a child with autism, what's the risk that their next child, or should they even have another child? And then last, and I wish we were farther along than we are, is that by identifying the genes, there's a large number of us that believe that we'll be able to begin to develop therapies down the road that will improve the quality of life of these children. So I don't have the illusion anymore of cures, but I do have the belief that by identifying the genetic causes, we will at some point be able to develop therapies, whether they're behavioral or pharmacologic, that will actually improve the quality of life for these kids. All right, so that's my 10-cent speech on why we do what we do. And now I want to share with you some of the results. So back in 2001, I'm going to argue that, in fact, our ability to essentially identify genetic causes of autism was really due to two major, what I think are revolutionary moments or watershed moments in our field. And the first, I'm sure you've heard of, probably at lectures like this, in this very auditorium, was the sequencing and assembly of the human genome back in 2001. So this was the more or less complete genetic blueprint of kind of what we call a reference human genome, which identified all the genes in our genome, where they mapped, where they were located on different chromosomes. And in our own case, we used this information. I was involved in sequencing the human genome. We used this information to predict areas of our genome that were going to be genetically unstable. So what I'm showing you in this complicated map is work from my student, one of my first students back in 2001, where we predicted areas of our genetic code. And you remember, our genetic code was just revealed to us really literally 6 months before, where we believed there would be genetic instability. So every area that you see in gold represent areas that we predicted in 2001, 2002 would be genetically unstable because of the architecture of these regions. And it's kind of, there were about, if you actually count the number of gold bars that are on here, there's about 111 of them. Everything with a letter behind it were things that we knew already were associated with developmental disabilities. So these are regions of the genome that were particularly prone to delete, so lose a piece of the DNA, or create an extra copy of it. We call that duplicate. And these areas of the genome we hypothesized would be important for explaining some of the unexplained cases of developmental disability. So the idea was pretty straightforward. We took the information from the Genome Project for these unstable areas, and then we systematically screened all of these cases. First it was 100, then it was 200, thousands, and then tens of thousands for deletions and duplications of these specific areas of the genome. So maybe I'll put this down so you can see it a little bit better. Were we successful? For over a period of about 6 years, our lab identified about a dozen new deletions and duplications associated with developmental delay in autism. So here's an example. These are children that have a specific segment that is relatively small. It's only about 500,000 base pairs, contains 6 of the 20,000 genes that we have in our genome. But these individuals are born, they have one normal copy of chromosome 17, and they have another copy that is missing these 6 genes. So it's one of those unstable areas of the genome. When we looked at these, what we found of these children, when we found this particular deletion, we never saw that deletion in normal individuals. When we went back and looked, we found that their parents were normal for both copies of chromosome 17. But what had happened, probably in the production of the sperm or egg that led to the formation of these kids, that piece of DNA was lost. Just a very small pocket of DNA with 5 genes in it. These kids are all unrelated to one another, but just looking at their features, you can see that they're similar. You see they have this kind of very characteristic nose. It's like what people have described as bulbous shaped. They have this protruding tongue, which is very common, and they have this kind of indentation in the upper lip, which we call that a pronounced philtrum. So all of these, essentially these kids, there are now 100 kids that have been identified with this specific deletion. And this deletion has never been seen in over 50,000 normals that we've screened. So this is like Down syndrome, like some of the other disorders, in the fact that it's actually missing as opposed to having extra copies of the DNA at this specific location. Here's another example. This is a deletion of about 3 million base pairs of the 3 billion that are in our genome. It contains about a dozen genes. You can look at these children. They all share one normal copy of chromosome 15, and they actually have one copy of chromosome 15 that is missing DNA from one of these unstable areas. So looking at those features of these children, you can see that they look similar, even though they're all unrelated. They have almond-shaped eyes. They have this kind of elongated and, in fact, kind of rounding of the forehead, which we call frontal bossing of the forehead. What you can't see from these pictures is these kids are in the 25th centile of growth. That means they are small children, even up to adults. So they're small children, and each one of them has a diagnosis of autism. This last example I'll show you is another region of chromosome 15 with a deletion that maps specifically at a different location, as indicated here. Clinicians looked at these photographs of these children, and they saw no obvious facial features. So these look like more or less normal individuals in the population. They all have mild developmental disability. But what was particularly interesting about this deletion is, unlike these two, which were always sporadic, we call that a de novo mutation. That is to say, they were always not seen in the parents, but they occurred, and we see them in the children. So presumably when the germ cells are being produced. These individuals, half the time were inherited, the deletion event, and half the time they were de novo. When we looked at the parents that were transmitting, the common feature among all of these individuals was that while the parents were normal in terms of their IQ and their development, all of the individuals that carried a deletion essentially were susceptible to epilepsy. So we had a seizure syndrome that was associated with this particular deletion that sometimes was also associated with developmental disability, sometimes associated with autism, but not always. So this is in fact important to think about, because genetic causes can be what we call syndromic, so that you can predict the outcome nearly with 100% or near 100% accuracy. In other cases, they can be much more variable. So sometimes an individual has epilepsy, sometimes the individual has epilepsy and developmental disability, sometimes the individual has autism. So the conclusion of that work and work from other labs, and this was done by a number of groups over a period of about 5 years, was that children with developmental disability and children with autism and children with epilepsy have an excess of large deletions and large duplications of DNA that are often de novo. That is to say, they're not inherited, and they basically, we call them collectively copy number variants. So I'll refer to them as CNVs as a shorthand for copy number variants. So large deletions and duplications of DNA. Perhaps most important, for the ones that we thought were the most severe, we did not actually see that often transmission. More often than not, these were actually sporadic mutations. So there are mutations that are occurring de novo, and that's why this individual would actually have the event, but none of the other siblings would be affected. So we call that a sporadic or a de novo mutation, because the event occurs before fertilization or immediately after fertilization. But importantly, this is not something that is acquired as a child ages. They're born with it, this susceptibility to essentially developing autism or developmental disability. A really interesting observation, and one that we've seen now multiple times, is that when these are inherited, because sometimes they are transmitted, like that one mapping to chromosome 15, we see that 60 to 70% of the time they are inherited from mothers as opposed to fathers. So somebody say, well, 60, 70 doesn't sound much. The expectation for a geneticist would be 50%, but this kind of departure from expectation is a big deal. So what does that mean? It means that for events that we think that are contributing genetically when they're transmitted, at least for these large copy number changes, mothers are more likely to be carriers of those events. And sons, males in particular, we believe are more susceptible to essentially manifesting autism and developmental delay, in part because females are probably buffered for essentially manifestation of these features. So, what are the lessons that we learned? Well, for a geneticist, this was kind of sobering, I guess, is that we were spending so much time thinking about inherited mutations, and what we were finding in these children with autism wasn't inherited mutations very often, but what we were finding these big deletions and duplications of DNA that were sporadic or de novo. So that means they weren't being transmitted, but they were happening when the child was being formed. Each of these was individually rare. So in terms of the proportion of intellectual disability or the proportion of autism that we could explain, each one was only a fraction of a percent. But collectively, it turned out that this accounted for a large fraction of cases. So when we did our math, what we estimated was these large deletions or duplications accounted for about 14% of children with developmental disability and about 7 to 8% of children with autism. So this is a big contribution in terms of actually the overall genetic risk. This feature, that when these types of events were inherited, they're more likely to come from mothers, is something that we've seen over and over again. And I think the other really important feature is that we were identifying new syndromes based on actually finding the genetic lesion first and then bringing those patients back into the clinic and assessing them in detail. So it's not as if, way we used to do genetics 10, 15, 20 years ago is we would argue that we have to do very careful assessment of the disorder itself, identify all the subtleties with it, and then gather those patients of that type. Here we were generally screening children with developmental delay and autism, identifying and proving a genetic cause, and then bringing those kids back into the clinic for a reassessment. So we call that a genotype first approach. So this was great. But we realized pretty quickly that this had limitations, this whole approach, at least this type of variation, because the CNVs that we were typically, or the deletions and duplications that we were finding, were typically large and involving dozens of genes. And so in many cases, we couldn't say that it was this gene that when you lost a copy was responsible for the disorder, or it was this gene that when you gained an extra copy, it was responsible for the disorder. And if you want to start thinking about developing therapies, you really have to get to the point of identifying the individual genes. So this is when revolution number 2 hit us. And I always joke with the students, I feel really lucky to have lived in a time where you can say that your field experienced 2 watershed moments. And I think this genuinely was a watershed moment for our field. And so the watershed moment was essentially something called next generation sequencing. So what I'm showing you here is one of these machines that essentially allowed us to sequence DNA or sequence genomes for a fraction of a cost and really at a pace that it was incapable of doing before. And so just to give you kind of a sense of how significant that change was, when we finished, or at least our draft version of the human genome, the estimates were that a human genome would cost about $100,000 or $100 million to generate. So this is, even for some of the richest people, typically out of reach. When the next generation sequencing technology came online, which was around 2000 and 2008, you can see the drop in the cost of sequencing a human genome. So this is, just to give you kind of a sense, this is actually orders of magnitude. So a 100,000-fold reduction in cost. Till today, roughly, it costs slightly more than $1,000 to sequence a human genome. So this is a big change, because it means that we can start thinking about looking at patients and looking at the genetic causes much more systematically. So that's basically kind of a comparison here for the rate at which computers have doubled their capacity or increased their capacity. So the reduction in the cost of sequencing has outpaced Moore's law, which we consider to be one of the big changes has been the increase in computational capacity. So sequencing capacity has actually been a much more radical change for our field. And so the idea was, well, let's go back to those individuals and let's actually sequence all the individuals that we didn't find a large copy number variation change or a large deletion or duplication. And so back in 8 years ago, this was still somewhat expensive. And so instead of actually focusing on the entire genetic code, what we decided to do was focus just on those portions that we knew were functional. Those are the portions that encode essentially the genes, which encode the proteins, which are the functional units which make cells work. So instead of actually taking on the entire genome and all the genic sequence, we focus specifically on the protein coding portions of each gene. So there's 20,000 genes, and we developed methods to essentially sequence about that 1.5% of our genetic code that encodes the functional proteins. And then this was the experiment. The experiment was, let's do the same thing that we've done before, where we're looking for large copy number changes. But now let's look for small mutations. We call them point mutations, just subtle changes in the genetic code. And the way we'll do this is we'll sequence families where we haven't found any genetic cause for autism, which was still the majority. And we will sequence a father, which is shown here by this square, a mother. We will sequence a child with autism, and we will sequence one unaffected sibling. So we call these quads. These families had in fact, all of these families had no family history of autism. And what we would specifically look for were the new mutations, the de novo or sporadic mutations in the child with autism, and we'll use the unaffected sibling in the family as a genetic control. Because this child doesn't have autism, and we'll compare the new mutations that we find in the children with autism versus the unaffected and ask if there's a difference. So this is important because what we were trying to do is identify the individual genes. By having complete sequence of those genes, it gives us the specificity to basically say this gene is a candidate, this gene is not a candidate. But we started initially by just sequencing these 2,500 families. Now this is still a large effort. It involves sequencing roughly 10,000 individuals from those families. And it's work that we did in collaboration with 2 other groups. So the types of mutations that we were looking for were really 3 types. So most of you should remember that your DNA that encodes genes actually produces proteins. So those 20,000 genes produce at least 20,000 proteins, and those 20,000 proteins are why you have a brain cell versus essentially a cell in your liver, because they're actually doing different things in different tissues to basically make the cell work. So there are mutations that have no effect on proteins. So basically they're silent mutations. And so those are interesting because they're kind of almost a control for these experiments. We have mutations that may, in fact, somewhat alter the function of the protein. So they often result a change of one amino acid to another, and we call those missense. And then the really interesting ones are the gene killers. So these are the mutations that break the function of a gene. So when you have that mutation, no protein is produced, or very little protein is produced. So it's the equivalent of completely losing that gene, just like that big deletion was that I showed you before. And so the idea here was to identify all the new mutations that were seen in a child and compare the pattern to the unaffected sibling in that family. So this is work from about 40 people over a span of 3 years, and we can summarize it in one slide. And my postdoc, I'm glad none of them are here. They're now faculty elsewhere, but it was a huge undertaking. And what I'm showing you here is basically the rate of new mutations that are seen in a child with autism, which is shown in red, versus an unaffected child from the same family. So first focus on the silent mutations, the ones that have no effect. You can see there's nothing different about the overall rate between the child with autism and the unaffected child. Okay? So there's nothing, we conclude, in fact, that there's no increase. There's nothing unusual about the mutation rate. So it's not as if autistic children have more mutations. But if you actually compare the truncating mutations, you can see that a child with autism is about twice as likely to have a gene-killing mutation than a child that does not have autism from the same family. If you look, this is highly significant and was observed by several groups around the world pretty much within about 4 or 5 months of applying this technology. The missense, these are the ones that result in a subtle change. There's a little bit of an increase in the autistic kids compared to the unaffected, but it's actually borderline in terms of its significance. So this was exciting because it suggested to us that, in fact, the model of finding that we had seen with the large copy number changes, those deletions, also applied to essentially the smaller events, the new mutations that were occurring. So a twofold excess of children with loss of function mutations means that in about 40 to 45% of cases, these mutations are responsible or are contributing to the risk of autism in those families. So it's a huge chunk of actually autism risk. We estimate this plus this difference accounts for about 21% of cases of autism. So that was the good news, an increase in the number of gene-killing mutations in children with autism compared to the unaffected. And these are just new mutations, not inherited. They're just basically occurring in the production of the sperm or egg. There was bad news. And the bad news was when these three groups actually focused on these families, we each did a different subset of families, and we didn't share essentially what we were finding because we figured we would find the autism gene. And in fact, we were really disappointed because in fact, we almost never saw the same gene hit twice. And back of the envelope calculation suggested that when we finally did compare all of our data for 2,500 families, that there were a minimum of about 500 individual genes. Each one is mutated can actually result in a child with autism. So think of it as 500 different roads to Rome. And in fact, at this point, because we had seen most events only once, we didn't know which of these events were responsible for autism. All we knew that was we had a very strong genetic signal for autism. So this was sobering because it meant that every researcher that had been working on autism for the last 20 years was woefully underpowered and would never have the ability by himself to prove individual autism genes. For a clinician, it meant that if you were seeing patients for 30 years, the chances that you had seen the same genetic cause of autism more than 2 or 3 times was close to 0. So we were shocked by it, but nevertheless went forward. There were other things that we learned when we were doing the sequencing of these families. One thing that we learned is we actually went back and asked the question, how many of these new mutations were arising from the father's side versus the mother's side? And what we found was that most of the new mutations, so those sporadic de novo mutations, they were originating in the father's germline. So about 80% were coming from mutations probably in the production or in the generation of the sperm itself. So 20% came from moms in terms of the de novo mutations, i.e. the egg, and 80% roughly from the sperm. We also looked at the average age of fathers, and we binned our patients into ones that had 3 or more mutations in their protein coding sequences, so those 20,000 genes, those with 2, those with 1, and those with none. And what we found was that there was a modest but really significant correlation with advancing paternal age. So in other words, as fathers got older, they were more likely to contribute more de novo mutations to their children. So I'm not going to spend a lot of time to go into this, but this has now been replicated and shown by multiple groups. And now at the genome level, we now estimate that for every person in this room, there are at least 60 to about 100 new mutations in you that isn't to be found in your parents. So it's not specific to autism. It's a general property of being a mammal, is that in fact, or even actually being a vertebrate, is that as individuals, particularly fathers, age, there's an increased risk of actually having these. 80% of the mutations arise in the father's germline. That means that old fathers are contributing more new mutations to younger children in the family than younger fathers to the firstborn. And this is also consistent with what we know about the epidemiology of autism. So on very large studies involving millions of individuals, people have found that fathers' age is in fact one of the biggest risks for autism. So a father that's roughly about 25 years of age, the chances that they will have a child with autism, particularly a son, is about 1 in 100. And when a father is 50, the chances that they will have a son with autism increases to about 1 in 60. But because we don't have family sizes of 60 or 100, it's usually a non-issue for most people that are over 50. But it still is a significant increase in risk. And we think that's because, in fact, there are more new mutations that are being contributed and more chances to break a gene, so one of those 500 genes. The second thing, and this is actually work we did with El Hanan when he was a young professor here, is when we actually looked at all the genes that were being hit. And remember, we didn't know which genes were causing autism and which ones weren't. What we found was that those genes that were being hit, in fact, were not just a random sampling of the genes in our genome, but they tended to be related to genes that did very specific functions in the developing individual. So what I'm showing you with this kind of complicated graph is every circle And the size of the circle represents mutations that we saw in a specific gene. So these are different gene names indicated here. So, for example, CHD8, we saw more patients. In fact, we identified 7 patients that actually had loss of function mutations, which is indicated here by red. Blue indicates these missense mutations. So these are the gene-killing mutations, and these are the missense mutations. And everywhere you see a joining line, that means that we know that, in fact, when these things produce proteins, that the proteins interact with one another. So genes just don't live and operate individually within a cell. They often work with other proteins. The products work together to actually do specific functions. And when we asked the question, what are those functions? What we found was that the genes that were being knocked out or killed in children with autism tended to relate to the function of neurons, particularly the function of the synapse, which is how we actually connect our neurons together. And interestingly, they were also associated with this pathway called Wnt signaling, which has often been implicated in cancer, but is generally associated with cell growth. And then a whole set of genes that are associated with chromatin remodeling. And these genes are actually expressed particularly early in development, and they turn on not tens, but often hundreds of genes at a specific time point. And they're thought to be particularly important very early in development. These genes that are involved in essentially the connectivity of the brain are actually expressed typically in the first year to 2 years of life at the highest level. So these are expressed postnatal or after birth. And these genes tend to be expressed very early, often 13 weeks post-conception. And in fact, if we project all the genes that we identified with a loss of function or a gene-killing mutation, something like 67% of all the genes could be projected onto these 3 networks. So why is this important? Even though we have this extreme kind of heterogeneity and 500 different causes, genetically they boil down to 3 or 4 pathways, at least for the most part, which is really important when we start thinking about potential treatments. So here's the problem. This is where we were circa 2012, 2013. 500 autism genes, and in most cases, we couldn't tell which of these genes, at least we estimated there were 500 autism genes, but we didn't know which of the genes we had identified were really causing autism. So there's a solution to this, at least one of the solutions, and that is to sequence not hundreds, but tens of thousands of individuals. And that would give us the power to say if we saw something 10 times, 20 times in our cases with autism and never saw it in our controls, we could be confident that we actually had identified a genetic cause. So there's a technology, and I had the good fortune of being in this department when a young faculty named Jay Shanduri came from Boston. And he had brought with him this technology that he had developed, which is called molecular inversion probes. And I'm not going to go through the technology other than to say that what this technology allows us to do is target a small number of genes and resequence them really in large numbers of samples with high accuracy, so exquisite sensitivity, exquisite specificity, and most importantly, we could do it for cheap. We could target 50 genes, at least these are research costs. It would cost us to target 50 candidate genes in a sample would cost us about $12.50 a sample. So if that exome that I was telling you about was poor man's sequencing, this was poor man's sequencing, because it essentially allowed us to really screen not thousands, but tens of thousands of patients. And so that's what we've been doing. We've been doing this for the last 3 or 4 years. And obviously we have to find tens of thousands of patients, which isn't necessarily easy, although we know autism is extremely common. Getting DNA for all of them is not. And so I've spent a lot of time over the last 3 or 4 years flying from one location to another, establishing a consortium of investigators who have children with undiagnosed causes of undiagnosed autism and undiagnosed causes of developmental disability. We put those all together, and over the last few years, we've assembled about 18,000 children and about 4,000 controls, typically unaffected siblings, where we can actually target the genes that we discover and try to prove that we've identified individual causes of autism, which is what the families really need to know. They need to know not these aggregate statistics, they need to know whether this gene, when mutated, is responsible for my child's autism. And the beauty of this work is that it involved essentially collaborating with about 13 different labs here in the US and about 21 different labs in China, with a lot of effort being done with Changsha University. And so just to give you the take-home message, this approach has been successful in the sense that of the estimated 500 genes or so, we think we now have identified about 250 of them. So we still haven't identified all of them that are contributing to risk of autism and developmental disability. So these are all the gene names, and whether it's a gene killing where the predominant mutations are gene killing or there's severe missense, it is indicated here in red and blue. The important point is the higher a gene is on this scale, the more confident we are that we in fact have identified a genetic cause of autism. So basically that means we've identified more families with the disorder. So what do you do when you identify now and you think you've identified a gene? Or of these 250 genes, about a third of them were previously unknown, to at least to the researchers that have been focused on autism. You bring those families back in for reassessment. And so this is what we have been doing with all the centers that we're collaborating, but in particular with the UW and Seattle Children's Autism Center, is once we prove a gene when mutated is resulting in autism, at least have the statistical evidence, we then bring those families back in one at a time for a clinical reassessment, typically lasting about 2 days. And why do we do that? Well, for a couple reasons. One, we want to make sure that we've identified the mutation, that in fact it is actually a de novo mutation as opposed to one that's inherited. But we're also bringing the patients back in to see if they are clinically more similar to one another than just run-of-the-mill autism, if that makes sense. So is there any features about these individuals with the same genetic cause that maybe give us a handle on what's going on with this specific form of autism? So you think of it as subtyping autism using genetics. And so these are one of the first sets of families that we brought in. This is this gene, which is involved in that chromatin remodeling I was telling you about. It's a gene called CHD8. These are the first families that came here back into the Seattle clinic. We screened initially 8,000, followed 13 of these gene-killing mutations in those 8,000, and 0 out of 9,000 controls that we screened. So you don't need statistics here to be confident that, in fact, we're finding something that is associated with autism. When we went back to the families, because remember, we were just screening the children with autism and developmental disability, 11 of the 12 are de novo. So these gene-killing mutations are, again, not being inherited. They're de novo, and in these families, the autism is also out of the blue, no family history. When we brought these kids in, including this one adult, 87% had a diagnosis of autism because some of the kids came in with intellectual disability without autism. But interestingly, and you can see it from these photographs, 80% of these kids actually had a larger head. So we call that macrocephaly if it's 2 standard deviations beyond the mean. You can see it in these pictures up here. When we actually have evaluated the clinical records of these individuals, 73% had severe gastrointestinal problems. So for example, some of these individuals, in fact, the stories were almost identical, although these individuals are not related to one another, of these families experiencing weeks where the child would have constipation, followed by weeks of diarrhea. And this would go on for years and years of the family's life. Interestingly, and this one hasn't reached significance yet, about 67% of these families reported pretty severe sleep dysfunction. So the sleep dysfunction was of the type where these kids would actually go often 2 days, 3 days without what one mother said is an iota of sleep. And she goes, and then they would crash. And in some families, they were actually telling us how they would actually lock the door from the outside because they were afraid that their kids would wander away in the middle of the night. So what do you do when you have these features, which we think are all relatively subtle, but maybe diagnostic of this particular subtype of autism? You mutate it in a model organism. So I had a postdoc that had just joined the lab that was familiar with essentially fish, particularly zebrafish. And what we decided to do was knock out this gene specifically in the developing zebrafish. So these are the zebrafish embryos as they grow. So we use two different types of technology. One is called morpholinos and one is called CRISPR-Cas, but the effect is about the same. We were able to either reduce expression or completely knock out expression of this gene, which is highly conserved between man and fish. So when we did this, and this is actually fish heads, what you can see here, these are the eyes, developing eyes of the fish. And what we found consistently is that this distance between the two eyes, we call these the eye cusps, was actually bigger whenever we knocked down this gene in the fish. When we determined what cells were responsible for this difference, what we found was that most of this difference was essentially due to an increase in the number of neurons. So these fish, when we knocked down this gene, actually had an increase in the number of neurons that were being produced. So if you think about it, this is reminiscent of what we saw in the patients. A knockdown resulted in macrocephaly, and one explanation could be an increase, not a decrease, in the number of neurons in these individuals. We also looked at the gut. And so what we did in these fish that we knocked out or knocked down the gene is we actually stuffed fluorescent beads in their mouth, and we measured how long it would take for them to poop them out. And so here are the fish, and we measured essentially distance from the mouth to essentially the anus in 4 segments. We did the control fish, and then we did the knockout and knockdown fish, and we did about 3 hours we did the measurement, and we did it at 6 hours. And so you can see at 3 hours, and then specifically when you move to 6 hours, most of them have gotten rid of these fluorescent beads, the normal fish. But in the case of the fish where we knocked down this gene, you can see that these fluorescent beads were getting hung up. They would get stuck in zone 2, zone 3, even at 6 hours. So we asked the question, why? And so many of you may not know, but in order for your digestive system to work, you also have neurons that are actually connecting with the brain to actually help with the movement of food in your developing gut. And when we stained for those neurons, what we found consistently is that the fish that were knocked down or knocked out for that gene had fewer neurons in their developing gut. So if you think about it, this is kind of interesting, because in the developing brain, the problem is having too many neurons, and that's what leads to the bigger head. But in this case, what we're having is too few neurons, which you think are actually resulting in this problem with that digestion. But importantly, we're using this organism to really prove these phenotypes, what we call these features of the disease, in essentially this model organism. So this isn't the only gene. So we've done now close to about 30 different genes this way. And I'm going to go through each one of these genes over the next hour. No, I'm kidding. I'm just going to show you two more quick examples. This is a gene called DYRK1A, which we found in significant excess. So 8 truncating mutations out of 7,000 children that we screened. 0 out of 6,000 controls. 7 of the 8 were de novo, all gene-killing mutation. Bring those kids back into the clinic, have characteristic features, 100% intellectual disability, 100% microcephaly, which means that essentially the head circumference is smaller than usual in these kids. So it's the opposite of what we just saw. 90% have late-onset epilepsy. 83% have an autism diagnosis. And the clinicians, when they saw the first 3 or 4 pictures, they said, oh, there's a common facial features here. You can see this kind of pointed chin in these children. You see this kind of long nasal bridge. And what you can't see is these children also have very specific foot abnormalities. So in this particular case, we didn't have to make a model organism. Someone had already made it. So they knocked out the gene, in this case, the mouse. And what they found is when they knocked out the gene, the brain was essentially smaller in size. So this is a normal brain in a mouse, and this is one where that gene is knocked out. And you can see the reduction in size. And these mice also had problems with behavior, increased anxiety, similar to what we were seeing in the humans. One thing that's interesting about this particular case is you can't see it from the pictures, but these kids also, because their jaw is so small, they have problems when their permanent teeth erupt. And in fact, most of them have to have teeth extracted during kind of pre-puberty to basically accommodate their smaller jaw. In fact, this gal here looks pretty normal. Actually had significant reconstructive surgery. This last example I'll show you has neither an increase in head circumference or a decrease in head circumference. So this is this gene called ADNP. So we call it the ADNP autism subtype. These kids actually look, to me, they're quite beautiful looking kids. They have normal, pretty much normal head circumferences, but when you actually study the photographs and you do measurements, what you can see is they have this kind of unusual rounding of the forehead. The clinicians call this frontal bossing of the forehead. 100% intellectual disability, 100% autism, 90% loss of expressive language. But 63% of these kids have an abnormal MRI, which we think is related to an overgrowth, this case specifically of the frontal cortex of these kids. We don't know a lot about this gene. It's still pretty much a black box. But we do know that when you knock it out, that in fact the neural tube fails to close properly in these mice. And we also know that in fact when you actually knock out just one copy, the mice have cognitive problems. So it's a pretty important gene early in development, and we find that kids that have this gene particularly mutated are at significant increased risk for autism and developmental disability. One thing I'll just mention is that one of the mothers of these children, once we identified the genetic cause, she became what we call in the field a lion mother. She went after to find every case of ADNP that was in the literature, reported or not reported. She contacted clinicians, contacted families, and she gathered all the information she could about these families. And I have to tell you, one of the big surprises for me as a researcher is the fact that when we start to connect these families that feel that they're so isolated, that when they find other families that have the same experiences that they have had, because they have the same genetic cause, they automatically connect in a very powerful way, and the social media has just made this so much more easy. In this particular case, when this mother started to look at the clinical records, what she found, she talked to the other mothers, basically. She found that there was a very specific feature associated with 90% of these kids. And the feature was that these kids had a full set of teeth by the age of 1. So it's something that none of us had ever bothered to think about or even consider, but this is where the mother actually beat the researchers, and she actually wrote the paper. Well, she helped write the paper, and she became the last author, which is like the honor position in a paper. It was just published last year. All right, so the final phase, and this is where we are right now, is that combining all these large deletions and duplications, which we call CNVs, and these point mutations, even the most optimistic estimates suggest that we can only account for about 30% of cases of autism. And I told you that 70% of autism should have a genetic component. So we have a lot more work to do. And so the things that we've been doing in my lab is actually going back to all those families that, in fact, we haven't been able to explain the genetic cause of autism, and actually subject them to full genome sequence. So remember I told you before we just focused on the protein coding bits. Now we're going back to those families and we're sequencing the entire genetic code. And we have a hope that we will actually find additional types of mutation, and in particular the mutations that we're interested in are mutations that affect not the protein coding portion, but the parts of our genetic code that work together to tell when genes are turned on and how they're turned on. So we call that regulatory DNA, because it's not DNA that encodes protein, but actually encodes when and where the gene is expressed. So I have a very brave and ambitious postdoc named Tashelle Turner who's worked on this now for the last 3 years. It's a real hard problem because we're taking those families which we consider the hardest cases, in which there is no obvious deletion or duplication, and there's no obvious gene-killing mutation. And we're saying, can we find any other signals that might give us a clue in terms of genetic cause? So she's basically just published a study where she took 516 of these hardest cases. So these are the ones that the clinicians would say, I have no clue what's going on. We sequenced 2,000 of those genomes, and they were all families of this type, one affected, one unaffected, a mom and a dad. And again, we're looking at new mutations, so de novo mutations, and we're comparing the pattern in the child with autism versus the unaffected. So these are preliminary data, so I'll just share them with you. What we've found, which we are encouraged by, is we find a trend. So these are different classes of that regulatory DNA. So we're just focusing on DNA that we think is important in terms of regulating the expression of genes. And what we see is a slight excess in the number of mutations, new mutations, in children with autism, which are shown in red, compared to their unaffected sibling. If we restrict that analysis to pieces of DNA that we know are in fact regulating the expression, and particularly early in development in the developing brain, this result becomes significant. And if we restrict the analysis to that subset of genes that we've already identified, so this is done at the entire level of the entire genome, the result becomes significant. So in other words, new mutations that are occurring in these families or in these children that actually affect the expression of these genes, so when and where they're turned on, seem to be an important potential cause of autism. The really interesting thing, and again, this will need replication, is that in these families where there is no obvious gene-killing mutation, but there is a mutation in a piece of DNA that in fact is important in regulating expression, we often find more of them in multiple genes in children with autism compared to their unaffected siblings. So it's not just one gene that's being hit. It's like lightning striking 2 or 3 times at critical portions of our genetic code that are important in regulating expression of genes. So we have seen in the first families that we've looked at, and we now have some replication of this, that in fact these non-coding portions of a genetic code are important, but you might need to have multiple hits in them to really push a child toward an autistic state. And so these are just some examples of families where we found either small deletions and point mutations that are coming in. So here's a family that these are 2 genes that people know are associated with autism already, and there are events that we're identifying, often multiple events. So they have 2 or 3 genes, but not the protein coding portions themselves, but the regulatory DNA. All right, so putting this all together, we think we are starting to get a handle on the genetics of autism. So the first thing I told you is that there is an excess of deletions and duplications, big ones, in the genomes of children with autism. They're about twice as likely to have one of these events than essentially an unaffected or a normal individual in the population. So many of these are sporadic, so they're de novo, but when they are transmitted, they're coming preferentially from mothers. Talk to you about a significant excess of gene-killing mutations in children with autism compared to unaffected siblings, and specifically de novo or new mutations. So not inherited from either parent, but essentially coming in from the germ cells, the sperm or the egg. And when we try to figure out where those come from, they come preferentially from the father's germline, 80%. And the older a father gets, the more of these new mutations he's contributing. And then last, this is not, in my mind, yet proven, but we think it's intriguing. We see an excess of new mutations in the parts of our genetic code that regulate the expression of genes. And often in children with autism, we are seeing multiple genes being affected as opposed to just a single gene. So we think this is consistent with a multi-hit model for the disease. So in summary, large copy number variation, big deletions, big duplications. You can think of them as like mini trisomy 21s in a way. They account for about 10 to 15% of cases of developmental disability and about 7 to 8% of children with autism. The sequencing of the coding regions shows an excess of these gene disruptive, particularly these sporadic mutations that are occurring, and then particularly subsets of genes that we know are important either in the development of the brain or essentially the function of the brain. So combined, we think that we have a handle on somewhere between 27 to 30% of autism, or at least the genetics of autism. And then I shared with you this idea, which actually started with the large deletions and duplications, is that once we prove a genetic cause, or we have strong evidence in support of it, we then bring those families back together, and we actually find features that previously we didn't recognize that help to distinguish that subtype from the other subtypes of autism. We think this is particularly important. Ultimately, we think important for essentially developing therapies for these individual families, because we think it'll matter what type of mutation and what has gone wrong in each of these individuals. And lastly, I showed you there's some preliminary evidence for an excess of non-coding regions of the genome being affected. They're often seen in multiple hits in these individuals. But if this is actually a common cause, and we still haven't determined how common this is, it's a minimum of about 5% of the cases, it means that every family with a child with autism deserves full genome sequencing. So no shortcuts. Basically, full genome sequencing of the child, full genome sequencing of the mother and the father to completely understand all of the mutations that are present. Because if it is multiple hits, simply finding one is not going to be sufficient to actually prove the cause. So I think it's a great time for autism genetics. I've been accused of being a half full as opposed to half empty kind of researcher, so I definitely believe the glass is more than half full. In terms of what we've understood in the last 10 years, I think it's transformative, at least from a genetics perspective. We really have handles on different subtypes of autism for the first time. This last part, this green part, which is the unexplained genetic risk, is going to be the hardest part, for sure. I think this is some of the easy stuff. And if it wasn't for the technological advances, we couldn't have done that work. The second thing, I guess, I've realized is that this is a global endeavor. Each of us is chipping away at one cause at a time, genetically, and that explains a fraction of % of cases. But for the families that have the CHD8 mutations or the DYRK1A mutations or the ADMP, it's the only genetic cause that matters. And the fact that we can connect them, I think, is a real useful application of genetic technology. Because long before we have cures, we were actually taking these families from a position of isolation and putting them together with other families. I can't tell you how many mothers have contacted me, after the fact, and even though I'm far removed and I always have clinicians buffering me, they still manage to get through, and they tell us how much it means to them to know why their child has autism. And we always feel apologetic about it. We say, well, there's nothing we can do yet. I mean, all we can do is identify what we think is a strong cause. And the answer we get from them is that just knowing that that it wasn't something I drank in my second trimester, right? That it was in fact something that couldn't be avoided, but it's actually valuable to the families as too, because if it's a de novo mutation, there's no risk for their children, other children, for developing a disease or for their children to have other children. And that, I think, is incredibly powerful information to give families. But the thing I think I've realized the most is that this can't be done by just researchers and not simply a small number. It involves connecting everybody in different centers around the world, clinicians, researchers, but probably most importantly, like the example of the ADMP, the moms and the dads, the families. And they in the past have been typically silent partners of research. And what I have seen more and more often is that they have often the critical insights for that specific subtype of autism because they're dealing with it on a daily basis. So I think this is where we should be. Clinicians and researchers should get off their high horse and actually work together with families in a tripartite relationship. And then lastly, the big hope is this. You could show this for 1,000 different disorders. Same thing could be shown for cancer. But this is the hope that drives the research. By identifying the mutations that affect the genes, we know the genes are producing proteins. Those proteins are working together to generate networks that function, provide the functional part of cells. Finding individual genes is just a start, because that gives us the diagnostics. So we can say something to the families about why and recurrence risk. But I think the point is not hopeless, because with 500 different genetic causes, if they boil down to a dozen different networks, I believe the treatments, when they develop, when they occur, will be at the level of the networks. And so it'll matter what type of breakdown you have in that child with autism. Is it a developmental gene that's expressed early, or is it one of these postnatal genes that is expressed, in the synapse later in life? Because I think the treatments will be personalized based on the networks. So there's a lot of work here. So if you're a young person and you're thinking about the future, you need to be thinking about how to tease apart these networks and how to activate or repress these networks, because I think that's going to be the treatments. So these are all the folks that did the work. I want to just shout out to 3. Brad Coe, who did most of the deletion and duplication, i.e. CNV work that I presented, to Shel Turner, who did a lot of the genomes data that I presented, and then Brian O'Rourke, who's now his own boss at University of Oregon Health Sciences, where he's a young assistant professor. really great collaborators at UW, particularly Jay Shanduri, Debbie Nickerson, who is like the muscle of all muscle when it comes to sequencing. She can do more sequencing than most. And then my colleague, he's the director at the UW and Seattle Children's Autism Center, or the Seattle Autism Center, Rafe Bernier, who's been critical for the recontact of the families. Lots of colleagues and clinicians. And then I love to show this picture, and I apologize to my lab because it's a little bit out of date, but I love it because it shows the sun shining in Seattle, which we've had a lot of this last month. And then I think most importantly, the patients and the families. Because without their contributions, and they have been active participants in this research, we couldn't do the work that we're doing. So we are eternally grateful for this contribution. And with that, I thank you for your attention.